Happy Wednesday morning. What we will do today is to go through the study guide for this week's exam 2. And that study guide will cover 3 chapters. So we will have a look of the chapter on heart, blood vessels, and also the lymphatic and immune system. Before we jump into the study guide, though, however, there's a couple of things that I really do want to quickly mention. But as always, our main focus on this session is to go through that study guide together. So towards the end of the last week, I started receiving some messages that students were having issues with accessing the Canvas and all the material there, or it wasn't stable enough to complete the assignments. So what I have done as a result of that, I have extended last week's assignments due dates until the end of this week. So you still have this week to get caught up on those if the Canvas gave you a little bit hard time last week. And on that note, it really is worth finishing off all those missing assignments, because where we are standing at the moment is that we're halfway through the semester by the end of this week. So once you have completed the exam 2 and lab practical 2, you really have gathered half of all the points for this course. We do, of course, have our finals week, and you remember that for my course on finals week, rather than doing an exam, there is a project that you have an option to complete as an extra credit assignment. So really, by the time we get to the end of the week 16, finish off exam 4 and lab practical 4, you are done with the material that counts towards 100% of the score. Then you do have a chance to earn a little bit more points to go beyond that 100% if you're not quite where you want to be with your grade. So that's your finals week is a chance to kind of adjust that. But really, I strongly encourage you finish off this week strong, and by the time you're finished, you are halfway done through the course. So it's definitely worth getting all those points. In general, on these online classes, hybrid classes, the key is to keep collecting those points throughout each week, so you don't have so much to do to bring up the grade on only with the exams. And that brings me to mention that this week there are no new assignments that I have allocated. I'm simply expecting you to complete the exam 2 and lab practical 2. This is a good week to get caught up. If you missed any assignment, or if you need any assignments reopened. So if that's the case, really for the first half of the semester, which will end at the end of this week is the chance to get those points, get those assignments taken care of. You do need to let me know. You do need to contact me, because many of the assignments do have a due date. That means that they get locked on Canvas once that due date has passed. Well, if you do not meet that due date and you still want to attempt that assignment, or if you did meet that due date and your grade for that assignment wasn't where you quite wanted it to be, this is the week when you will reach out to me, get the approval for it, and I will extend your due dates. I will extend them only until end of this week. So the goal is really that by the time we move on to the next week, we can say that we have wrapped up and finished the first half of the semester. That way also your grade reflects accurately where you're standing, and that way no one finds themselves in a situation that at the last weeks of the course you'll be cramming to get through any missing or oversights of assignments. So this week, really, and I do have here a little bit of a correct immediately. Of course, we will be tackling the exam 2 and lab practical 2. And both of those are due at the end of the week. So on the October 17th, I believe is the date, and 1 minute before midnight. What you'll notice is that exam 2 really carries quite a bit of points. We're talking about 100 points towards your final grade. Lab practical, half of that, 50 points. Since we did the first lab practical, you remember that there were a little less questions because we had not covered yet as much stuff that I could really pull those questions from. So there was not that much to test. Now the week 2 and then on the coming weeks, we actually have a little bit more of a material to test. So lab practicals will be also a little bit more substantial. In terms of the format of the both of these for the exam, you remember the material is from the lectures and lecture videos. You have lecture videos available on Canvas. We will go through a study guide, and every question that I ask in the exam portion is from that study guide. However, the study guide is only partially completed, and this session here, in addition to our session on Tuesday and on Thursday when we meet in person, plus these Wednesday sessions when we meet virtually, these are the only times that I will be going through the missing bits of the study guide. So there will be no published complete study guide there. Study guide that I have shared on Canvas already, that does have some gaps. And usually those gaps are important topics, important keywords, important concepts that I really want you to be familiar and comfortable with. So that's part of the pedagogical approach, but that's also part of the responsibility of the student to attend to these sessions, since you can find so much of the answers on the study guide. Otherwise, it would be me giving you answers for the exam. So I hope that that makes sense. And that is really what we will focus on this session. We will go through the study guide, and I did this in a class with you on last Tuesday or yesterday, and we were able to go through the entire study guide in an hour and 50 minutes. I am going to be honest, it is a little fast-paced to be able to accomplish that sort of a speed. So just be aware of that while we will be going through it, we won't have a chance to go through in detail of the nitty-gritty. That has been covered in the lectures, that has been covered in the lecture videos. So I'm expecting that if you are feeling uncomfortable about any of those, you're able to refer back to those. This today really is about making sure that you get a chance to get those missing answers. It is a review, it is a general overlook of the things. So if there's any gaps after that, you will have to go back a little bit. Of course, as always, I am available for one-on-one or small group meetings if you do want a guidance as you're revising those topics. I strongly encourage you to also, before you tackle the lab practical 2, to use the practice version of the lab practical. And this is because this practice version shows you all the questions that come up in the actual lab practical. And I encourage you, since these questions are pulled from a larger pool, to work through that practice a couple of times so you have had a chance to see each one of those questions that might come up. Of course, there are no points associated with the practice practical, so it is up to you. If you feel that, not quite, I don't need that, I feel comfortable just jumping into the actual lab practical, go for it. But if you feel that you want to see what might be coming up, what you might expect. Well, this is a good chance to do that. So work through the practice version a couple of times so you get a chance to see as many of those questions from that bigger pool that might come up. And I sometimes get students asking for lecture handouts. You do know that I have uploaded those. So for each of the chapters that are covered on our course, there are the lecture handouts. These are from the textbook that your publisher uses, or our publisher's textbook. So it's the very same textbook that you will be using. They align very closely with the lecture videos, with the lecture slides that I use in my own videos or in the classroom. There is a little bit of difference in just the way I lay them out, simply because my lecture videos are kind of copyright independent, so they are not tied with any particular textbook. So that's the biggest difference, but really the same content is covered. So if you feel that those would be helpful for you, I by all means encourage you to use those as you revise the material. At this point, I'm going to leave the question a little bit till later. And what I'm going to do instead, I'm going to jump to the study guide and we'll have a look of that together and cover any of the missing parts of that study guide. So let me just pull up a right window. And as before, as we have done earlier on, we will work through each of the chapters that are covered in this exam. And as you remember, exam 2 covered the heart as a topic of its own. Ooh, I didn't mean to get it highlighted quite that way. Let's see what we can do. We're going to cover the heart as a topic of its own. And we will also talk about the blood vessels. You remember there was quite a bit of material that got covered when we talked about the blood vessels, especially when it comes to the anatomy. They are very varied, and really the best way to tackle them is just simple memorization. And the last chapter that we had a look of together was a combination actually of lymphatic and immune systems. So that's the third chapter. When I look at the study guide length is about 35 pages. That is largely because there are a lot of diagrams, especially when it comes to the blood vessels, so you have a reference to go back to. But when I look at how many pages have I dedicated for the lymphatic and immune system, well, lymphatic system has 5 pages and immune system 1.5 pages. That is not to say that these are not important systems. All of these are important, but the emphasis will be on the heart and blood vessels. There will be some questions about lymphatic and immune system, but it is definitely heavy on the 2 first chapters. So what we're going to do, we're going to start with the heart and just some of the basic things that we want to remember when we talk about the heart, and science that relates to that is that you probably would not go and see a heart doctor. You would go and see a cardiac doctor. You don't go and do heart science, you go and do cardiac science. So that's the proper medical term that we will be using and that we will be referring to. In terms of the heart and kind of mapping it, putting it on the map with everything else that we have learned, we have talked about the circulatory system in large. And I think that this is a good time to kind of remind us about all that and how that all relates. And let me just get the colors changed back so that we are on the right page here. So one of the things that we talked about was that how really cardiovascular system was almost like two hearts and two loops working together. So if this here is our heart, we have two circulatory systems that we can take in a larger scale out of that we need to know. First of all, we have blood going to the lungs. And this blood is deoxygenated when it goes to the lungs, and it's oxygenated when it comes back from the lungs. This first loop, if you wish, is what we know as pulmonary circuit. So the function of this circuit is really just get that blood oxygen rich once it has gone there. One thing that's quite interesting when it comes to this circuit is that this is the only time really when we see arteries carrying deoxygenated blood. So it's a bit of an oddity. Normally, you remember arteries always carried oxygenated blood. So the pulmonary circulation, the pulmonary circuit is the oddity where we have these kind of flipped other way around simply because now the blood that's leaving the heart is oxygen poor and it gets back to the heart after being oxygenated in the lungs. Well, the second part or second loop, if you wish, that we have really covers rest of the body, all other parts. So other parts than the lungs. And this loop carries oxygenated blood away from the heart to the tissues and then back from the tissues, back from the body as deoxygenated. This is known as our systemic circuit. So this systemic circuit carries the blood to all parts of the body with the exception of the lungs. So these two would be worthwhile just to have a handle on, have an idea of, because they are a big separation between the two loops that we find. So I'm kind of expecting that you're comfortable with that already at this point. But if you were not, this was a good chance for you to kind of have a reminder of. And really, today's focus on this chapter that we had a look of was, of course, on the heart. So heart was this driving pump that drives the blood within the blood vessels to these two circuits. And we're going to look at different parts of the heart, how this all works, and how these parts of the heart come together. Before we get too far, I think it's always useful to kind of let's define where we really are in terms of the organ when we discuss it in terms of the anatomical location. So what I have here is going to be just a really simple sketch of a human body, and we're going to locate the heart. Where is it within there? And we can divide this human body into half, of course. So we do have our chest cavity, our thoracic cavity. So heart is located within thoracic cavity. You might remember that at the very first chapters of BIO201, we went through all of these different cavities that the body is made of. And we're now going to be focusing on the thoracic cavity, the chest cavity. Within the chest cavity, of course, we have quite a few structures that we might want to use as a reference when we locate the heart's location. So we, of course, have the lungs. So the heart is located between the lungs. And you remember that the right lung is a little bit bigger, left lung a little bit smaller, and the reason why the left lung was a little smaller is because heart does take some of that space where the left lung would otherwise be. The space between the lungs that you sometimes hear referred is the mediastinum. So that's where the heart and some of the major vessels are located. We do have a sheet of muscle, thin sheet of muscle that separates the thoracic chamber from the abdominal chamber, or thoracic cavity from abdominal cavity. This sheet of muscle is, of course, known as our diaphragm. So heart would be located superior to the diaphragm. I realize and I see that on this version of the locational terminology, I have been a little bit easier. I haven't used full on proper anatomical medical terminology, but I do expect that you're comfortable with those since we have covered them before. Another way how we can locate the heart, which is usually quite useful as you are looking at the location of the heart from the surface anatomy, is by placing it based on the location in relation to the ribs. So we do have our rib cage, and heart is located somewhere between second and fifth rib. So that's usually a generally good way of where to start looking, especially when you're looking with a stethoscope, where to start really expecting to find the heart. So typically between second and fifth rib is where the heart is going to be located. Another thing that you'll notice is that the heart is just slightly off from the midline of the body. So just slightly to the left. Usually students, when they first look for heart without much of a knowledge, they go all the way to the left side of the chest. And that's too far off. Heart is fairly middle, just a little left. And one of the parts of the heart that we discussed that how heart was this kind of a cone-shaped structure, if you wish, how the very tip of the heart was known as apex. So many structures will have a part known as apex. It's the sharp end, the tip end. It doesn't always have to be pointing downwards, but of course, in case of a heart, it's pointing downwards, so inferiorly, and slightly to the left. So here. And this is really what gives our heart this axis that we can look at heart as independent from the rest of the body. And we use this as the kind of a plane, the long axis of the heart that we relate the terminology when we discuss heart independently. So that should put us to quite a good place of understanding where the heart is located. Other thing that we notice is that the heart wall itself can be divided into different layers. And let's have a look of that a little bit. So what I'm doing here, I'm drawing just a cross section of the heart. So I'm not drawing the entire heart. We're just looking of a small layer of the heart. And what we'll end up noticing is that there's actually not all of the heart is the same. There's 3 layers of the muscular layer, if you wish, the meaty part of the heart that we will notice. And we will do this journey of ours of looking at different layers of the heart in an order that will start from the inside of the heart, and we will work our way outwards from there. So layer after layer. So we're going to start with the muscular wall, and then we're going to continue by seeing other parts that surround the heart as well. So starting from the insides, so the very first layer, if we work our way outwards from the heart's center, the very first layer that you find that's facing towards the blood that's inside the heart is going to be our endocardium. So endocardium, the innermost layer, which we could see here. So directly facing towards the chambers inside the heart, directly facing towards the blood that's within these chambers. After that, we do have a thicker layer, and this thicker layer is really the layer if you're really wanting to study the heart muscle itself that we often end up studying. That's known as a myocardium. So because taking images of the heart, for example, is so difficult and heart is constantly moving, and especially if you do research like I've done quite a bit on MRI imaging, what you'll end up noticing is that it's hard to grab only the endocardial layer of the heart. So often we focus on the myocardium because we can be clear, we can be certain that that is the meaty bit of the heart. So much of my research has focused on the myocardial properties. After myocardium, the outermost layer of the heart, the layer that's facing out of the heart, another thin one is going to be our epicardium. One way to think of the epicardium is if you think of the earth, and we of course all know that we get sometimes earthquakes on the earth. When we get an earthquake, it never really occurs at the very surface. It occurs further into the inside of the earth. If it occurs here, the spot that's directly above the earth or on the surface of the earth, directly above where the earthquake took place, is known as epicenter. So that might help you to remember the epicardium term as well. I don't know if it doesn't, don't let that confuse you. That's just one way to look at it. The other thing that we end up noticing is that the heart itself is enclosed inside a fibrous sac. This fibrous sac is made of 2 layers, and in between these layers we have fluid. The sac is known as pericardium. And pericardium, like I said, is made of 2 layers, and we're going to have a look of those. So the first, the innermost layer, the layer that's immediately next to the epicardium, is going to be known as our visceral layer. The outer layer that's further away is known as our parietal layer. And in between these two, we have this space that gets left. And this space contains a great amount of fluid. And this fluid serves really 2 different purposes. The space itself is known as pericardial cavity, and pericardial cavity, and especially the pericardial fluid within there, helps our heart to have a lot of movement. If you think that think of the heart, it's located here between the lungs, so it would be easy for it to be constricted and not have room to expand and shrink. But by having this pericardial sac, we have a little bit more room for it to move and extend and shrink as needed. The other thing that the fluid within this sac allows, it allows less friction as these 2 layers of the pericardial sac slide past each other. So it reduces the friction as well. So one way to describe it is that it does lubricate these layers so they can slide easily past each other. So at this point, we have had a look of the heart, the different layers, working our way from the inside towards the outside. We have also located the heart. Few more things that I want to mention about the sac, and we kind of touched upon on these already. We talked about how this allows the movement. The other thing that the sac allows. And we've seen that many structures in the body are enclosed into these compartments. And these compartments were useful if we have an inflammation somewhere. The inflammation, if it's enclosed within a compartment or outside the compartment, it cannot spread quite as drastically. So really, that's the other good benefit that we get from placing structures within compartments. Of course, since we are talking about inflammations, it would be nice to do a little bit of medical terminology. So you remember whenever we had an itis, it was an inflammation of something. And in this case, if the pericardium is inflamed, we talk about pericarditis. We can hear this with a stethoscope as we listen to heart sounds. There's kind of this crackling sound as these layers don't slide as nicely past each other anymore. There is a risk with excess fluid building up and the pumping ability of the heart being compromised as well. So we don't want that happening. We need to be a little bit more aware. We have talked about general muscle types in the past, how there are in general three muscle types in the body: skeletal muscle, smooth muscle, and cardiac muscle. Well, we're going to be now shifting our focus on the cardiac muscle since that's the topic of the day. And just as a really quick reminder what a cardiac muscle cell looks, I've asked you here to circle the terms that apply to it. So first of all, we see that cardiac muscle cells are typically branched. So they have multiple endings. We also end up finding one central nucleus, typically in a cardiac muscle cell. We end up finding striations that run through the cardiac muscle cell. So those stripes. And we end up finding at these ends of a cardiac muscle cell these intercalated discs. These were structures that helped with passing material and messages from one heart cell to another. So really, if you think about it, the main function of the heart and heart muscle, heart tissue is that we get one synchronized contraction of this muscle. If each of the cells would be doing their own business, we would not get this synchronized contraction. So for that reason, we need to have these cells connected to each other. We need to have these cells really working in uniform, and these intercalated discs play an important role. A couple of other things to notice about cardiac muscle. Of course, we find it only at the heart, and it is not under voluntary control, so it's involuntary muscle. So you don't have to think every time that the heart contracts that, oh, I'm gonna have the heart contracting now. That would be problematic, especially when you're sleeping, for example. What I've done next, I've talked you through some of the major parts of the heart, major vessels that we need to know as the blood flows into the heart, through the heart, and back to the heart. And this kind of refers back to what we have already seen when we talked about the pulmonary circuit, so to the lungs, and systemic circuit to the rest of the body. So how I'm going to discuss through these parts is by using the most simplest way of drawing the heart that you can have. And you remember that was a box that we can divide into 4 chambers. That is really all that heart is in terms of if you want to consider the chambers. Of course, heart really anatomically is not a box, but we will start with this box diagram and then expand it to look at the actual shape of a more anatomically accurate representation of the heart. Let's have a look of the flow of the blood through the heart. So blood comes to the heart through superior and inferior vena cava. Superior vena cava drawing the blood from the upper parts of the body, inferior vena cava from the lower parts of the body. This blood arrives to the right atrium. So you can, in the spot test, in the practical, or in the exam as well, you can write it out as right atrium, or you can also use abbreviation RA. I'm happy either way. I will figure out what you're meaning. It's not going to be a problem. Once the blood has entered to the right atrium, it needs to then travel next to the right ventricle. But to travel to the right ventricle, we have there a valve that separates these. And the valves had a purpose that they facilitated only one way flow of the blood. So this is the method that we use to make sure that the blood can go only to one direction and one direction only in the heart. So the blood always flows from the right atrium to right ventricle, never to the opposite direction. And this is thanks to this valve arrangement. Well, this valve that we see is going to be called either as a right AV valve, so AV stands for atrioventricular, or sometimes it's described as a tricuspid valve because of the three leaflets, three cusplets that make up this valve. Other thing that's interesting is that if we look at the wall of the heart drawn here and this valve, the right AV valve, there are chordinae tendinae that connects to the papillary muscles to prevent these valves from flapping opposite direction from where they should be. So it's kind of a security mechanism there to ensure that the valve remains in the shape that it needs to be. Well, once the blood has now passed from our right atrium to right ventricle, the next step for the path of the blood is for it to leave the right ventricle and shoot out of the heart to our lungs. And there's going to be pulmonary trunk that leads to the lungs, which is where the blood goes first as it leaves the heart from the right ventricle. I do want to highlight from this diagram that, of course, the blood doesn't go from right ventricle back to the right atrium. The blood would be flowing behind the heart on a vessel or within the heart on the surface of the heart on a vessel. So it should be probably a dotted line that I've drawn there. I've just cut some corners there. Well, I didn't need to do that. Well, if the blood is going to be heading to this pulmonary trunk and then to the lungs, it has to go through one more valve that separates the right ventricle from the pulmonary trunk. And you will probably not be too surprised to hear that we have named this valve based on a structure that follows it. So it's going to be pulmonary valve. So by this stage, if we have looked at the diagram and we relate it to everything that we have written out here, we are now comfortable with the right AV valve, tricuspid valve, and also the pulmonary valve. And blood now goes to the lungs. It gets oxygenated there, so we get more oxygen added to this oxygen poor blood, and it returns back to the heart. And where it returns back to the heart is to the left atrium. And again, now blood has built up to the left atrium. The next step will be for the blood to move to the left ventricle. But again, as before, we want this blood to flow only to this direction and not backwards. So we have valves located here as an adaptation to ensure that the flow of the blood moves to one direction only. And there's a few different names that we can have for this particular valve. We can call it left AV valve, left atrioventricular valve, as we saw on the other side, now just with the right side terminology being used. Another term that we sometimes see being used to describe this valve is a mitral valve. And bicuspid valve is the third name that we can see. The bicuspid is a good way of separating it from the tricuspid, because that tells us that this valve, this AV valve, has only two leaflets, so two cusplets. So if you're comparing the anatomy of these, wanting to make sure that you can tell one apart from another one, it's really looking at the number of these leaflets that comes important here. So now we have gotten the blood from left atrium to left ventricle, and that was thanks to using this valve that had many different names: the left AV valve, mitral valve, or bicuspid that we could use. So that's something that you might see being used. Well, now blood has to leave the left ventricle, and it's going to leave from the left ventricle and then go to aorta. And as you might remember from our revision of the major blood vessels, that aortic arch had its own branches, and then aorta continued to the rest of the body, supplying this oxygen-rich blood so that the tissues could receive it. So again, the blood does not return back to the left atria. The vessel is rather at the front of the heart from the left ventricle. So I'm kind of drawing it as a dotted line, but there is going to be a valve that separates the left ventricle from the aorta. And not surprisingly, now that you know the naming convention, the name that we're going to be using there is going to be aortic valve. So aortic valve separates the left ventricle from the aorta. And now blood then continues to other parts of the body, rest of the body. The oxygen gets picked up and it returns back to the heart through these superior and inferior vena cava, which is where our journey started at the very beginning of our discussion of the flow of blood through the heart. So at this point, we have a fairly good understanding of the flow of blood, some of the important structures of the heart. I want to add one more structure. I typically like to ask this in an exam because it's a good example of terminology. And this structure is going to be interventricular septum. So septum was a wall anywhere in the body, separated some parts. And if it's inter, it means that it's between the following two structures. And of course, in this case, it's going to be ventricular. So between the two ventricles, the wall between the two ventricles is going to be called interventricular septum. So that's a structure that sometimes I do ask in an exam. So you might kind of want to make a mental note of that as well. Well, let's step onwards and have a look of everything that we just talked about, but put it on a bit more anatomically accurate representation of the heart. So let's start first, rather than jumping into the flow of the blood and all that, by highlighting some of those anatomical landmarks that we have just talked about. So interventricular septum separates the left and right ventricle. This wall here. We also talked about the apex, the very tip of the heart, when we were locating where the heart would be located. And that allows us to follow the flow of the blood through the heart. And I'm going to go through a little faster since this should be familiar. So blood comes through the superior and inferior vena cava to the right atrium. From right atrium, blood travels to the right ventricle, but these two were separated by a valve. And you remember that this valve that we will see here was called our right AV valve, or it could be also described as a tricuspid valve based on the three leaflets, three cusplets of this particular valve. Well, from there, blood flows from the right ventricle to our pulmonary trunk. And pulmonary trunk then went to lungs. So this blood now that was oxygen poor gets reoxygenated within the lungs. One more important structure that we of course want to notice is separating the right ventricle from the pulmonary trunk, and that's going to be our pulmonary valve. So another valve there. And these valves really facilitated that the flow of the blood is going to be to one direction and one direction only. Well, now our blood has been oxygenated. It's rich in oxygen, and it returns to the heart from the lungs through pulmonary veins. There's actually 4 pulmonary veins. I've just illustrated one of them here. And it is to the left atrium where the blood returns, and from there it has to move on to the left ventricle. Well, again, we want the flow of the blood to be unidirectional, so only to one direction. And how we are going to facilitate that is by having a valve. And this valve that we see here separating the left atria from left ventricle, it has actually a bunch of names. It could be called left AV valve, left atrioventricular valve, or mitral valve, or bicuspid. So all of those names would be fair use, would be describing the same structure. In an exam or in a practical, if you're being asked to name a valve, if you provide one name, that's enough. If you provide multiple names, well, that's even better. I'll give you extra credit for that. Well, blood has to leave from the left ventricle to the aorta. And you remember aorta then went to the rest of the body, so that this oxygen-rich blood could be spread there. Well, to go from the left ventricle to the aorta, we want to again make sure that the flow of blood is unidirectional, one direction only. We are going to go through another valve. This valve is going to be called aortic valve, following the structure that's located immediately after it. So I hope that that makes sense. We have moved from this very basic diagram to this a little bit more advanced diagram that's more anatomically correct. We can start to see some of the anatomical adaptations of the heart as well here, such as comparing the thickness of the ventricles and noticing that right ventricular wall is much thinner than the left ventricular wall. We have discussed all that in our lab when we did the heart dissection. So none of that should be groundbreaking new information. Remember these review sessions, we only focus on the key concepts. We won't get through all of the material that we have covered so far. We should talk a little bit more about some of the other terms that we have already, some of them explored, some of them we haven't, but I want to make sure that we feel comfortable with all of those. So I've shared with you about the valves, how their main function was to make sure that the flow of blood is one directional. The valves opening and closing of them is driven by the pressure. So as pressure increases on one side, the valve either can open or it has to shut close, depending on what side of the valve the pressure is. The goal of the valves really to make sure that the flow of blood is to one direction only. If there is failure in the valve of closing, we do end up getting some backflow of the blood, and that would of course make the heart to pump kind of back and forth the same blood. Heart doesn't work as effective. We can also have that the valves don't fully close so they are narrowed, so they don't open also properly, so we can have a narrowed valve, so that would be another clinical issue. Terms that you saw when we did our dissection of the heart, we talked about auricles as these kind of extensions of the atria. So they were flappy when they were empty, but it allowed atria to grow much bigger in size. Trabeculae carneae was this ventricular wall characteristic that we saw, that the ventricular wall was typically very bumpy in its structure. This bumpiness of the ventricular wall was important because it helps us to make sure that no blood stays behind there when the ventricle squeezes, so that it has a chance to stay still and pool and start to form clots. We have talked about the septum, especially the interventricular septum, so a wall that separates parts of the heart. Sometimes we might have dysfunctional valves, as I mentioned, and we might need to even get to the point where we have to consider replacing these valves. We can replace a heart valve by a mechanical valve, which these days is not that common, a popular answer, because anything that's mechanical has a chance of eventually failing. Everything wears down, there's wear and tear over time. So living origin is usually a little bit better. The other issue with mechanical valves is that they are one size and they remain on that size. So you can't put them on very young patients, because young patients' hearts will still keep growing, but the mechanical valve would not be able to keep up with the change in the size. We can get valves from dead humans. These would be allografts. We, of course, cannot take a heart valve from a living human because, well, we all need our heart valves. We don't have spares to give. So that is one donation that we look for when we collect organ donations, heart valves from other individuals. You can also, if we don't have human donors available, go for animal origin valves from animals. Pigs are a fairly good source of heart valves. So these would be our xenografts. Couple of other structures that I do want to talk, and these structures are really kind of more related to the embryonic development of the human being. So before you are born, there's really no way for you to do breathing. There's no reason for you to pass the blood to the lungs, since lungs are dysfunctional. That would be kind of waste of resources. So what we have this bypass that the blood goes directly from right atrium to left atrium before you are born, during your embryonic time. This passageway is known as fossa ovalis. So this bypasses the pulmonary circulation. Fossa ovalis closes immediately when you are born, so that the pulmonary circuit now gets recruited to work. Another similar adaptation is ligamentum arteriosum. That's another embryonic remain to bypass the lungs. So if we do end up to the pulmonary trunk with some blood to the pulmonary trunk, that can be also returned back to the heart. These are things that really play a role only on the development before you are born. One more structure that I do want to kind of point out. We talked about the coronary circulation, and we will revisit that in a second. How blood gets supplied to the heart muscle itself. Well, that blood needs to return back as well to the heart, and that's through coronary sinus. So within the right atria, we will find coronary sinus, which is an opening for the blood returning from the coronary circulation to the heart itself. So that's another structure that might be worth of keeping on an eye for. When we look at the surface anatomy of the heart, that helps us to work out the coronary vessels as well. Some of the basic terminology, you've heard a term sulcus, as this kind of depression on the surface of something. I think that's when we talked about the brain surface structure. So that might be familiar from their similar term groove as well. So there's three main depressions on the surface of the heart. We have our anterior ventricular groove that separates the atrioventricular groove that separates the atria and ventricle, also known as a coronary sulcus. So if I draw a really quick sketch of the heart from the anterior side here, we have our atria and we have our ventricles. And here we have our right side. We have our superior vena cava, inferior vena cava. We have our pulmonary trunk coming out from the right ventricle, and then we have our aorta coming out from the left ventricle. So there's the general sketch of the heart as it appears from the anterior side. But what I want to highlight is these grooves and sulcuses. So the atrioventricular groove, or coronary sinus as we call it, separates the atria and ventricle from each other. We also have something that we call interventricular sulcus, and we find it both on the anterior and posterior side. Interventricular means between the ventricles, and sulcus is depression. So we find it both on anterior and posterior side. And these are important depressions on the surface of the heart as they allow us to position the coronary arteries so we can then find them laying in these grooves and sulcuses. And let's go through these quickly, and we're going to start with the left coronary artery, which divides almost immediately after it has arisen from the immediately after the aortic valve. It divides into anterior interventricular artery, or sometimes called in more of a medical language, left anterior descending LAD. So it divides into this artery that runs down between the two ventricles on the anterior side. The other branch of the left coronary artery is going to be a left circumflex artery, and it runs in this groove between the atria and ventricle on the left-hand side, then arches around the heart and continues on the back side. So both of those would be arteries that are worth of note. Well, we are left with one more artery. This is going to be, of course, our right coronary artery. And right coronary artery runs on the right side from the after aortic valve and within the atrioventricular groove continues to the back side of the heart there. So these would be the major cardiac vessels that I do want you to be somewhat familiar with. We don't need to know the veins at this point, but do know these arteries. We remember that the veins were less regular. They collect the deoxygenated blood, bring it back to the heart, to the coronary sinus, to the right atria. And we saw the coronary sinus here when we discussed that structure. Now I think that we're at the point where we can start looking at the physiological aspects of the heart. Let's start with the basic heart rhythm. So we remember that the heartbeat is involuntary. Your heart will always beat. You don't have to think about it. And one of the things that we do end up finding when we look at the basic heart rhythm, we do notice the membrane potential of the heart resembling quite largely of the action potential that we saw in the nerve cells. This time it's just kind of all tilted to the side. So we have our pacemaker potential, the resting potential. It is increasing over time a little bit until we reach the threshold where the depolarization happens. So depolarization, we are going towards the zero, and then repolarization after that away from the zero. And we start the process again with the pacemaker potential. One of the things that I do think is really important for us to be familiar is the path of how the heart muscle and heart tissues, messages within the heart, the electrical conduction of the heart, how it propagates through the heart. So let's have a look of that. It all starts from the sinoatrial node. which is really the pacemaker of the heart, generates the heartbeat, if you wish. And from there, these electrical impulses spread through both atria on the right and left side, passing that electrical excitation, being excited through both atria. Then we reach the AV node, which is kind of a delay station, relay station. So we take a little pause there, giving enough time for the atria to contract so that the blood can flow out of them. And only then we continue that message. We're really trying to kind of slow down the progress a little bit so that the blood has time to move from one chamber to another. Well, signals travel down the bundle branches, bundle of His and Purkinje fibers, especially at the interventricular septum, and then continue arching around to the ventricles, causing now ventricles to contract and push the blood out of them. So really heart kind of beats in a rhythmic phase so that either atria or ventricle are contracted so that the blood can flow from one to another. We need to have a little bit of time for the blood to travel from one to another. So let's have a look at what else we could talk about. Some clinical considerations as we're talking about these electrical conductions. If we have irregular heart rhythm, so the heart rhythm doesn't follow what we would expect it to be, we end up with the arrhythmia. Arrhythmia referring to the irregular heart rhythm. One example would be when the heart can go into fibrillation. In fibrillation, we have really fast, irregular contractions. Now the heart is not as controlled, is not as synchronized in passing the electrical current, so the blood doesn't flow in a controlled manner. We're not really able to pass the blood onward, so it's just all over the place, going crazy. So what we can do if we have still some sort of rhythm that, even though it's rapid and irregular, we can defibrillate it. So the defib, automated external defibrillator, AED, is what we use. So we give the heart a shock, trying to interrupt this chaotic contraction and re-establish that normal rhythm. So that's what we do there. Some people have this happening frequently, that their heart goes out of the sync, and they might, rather than we administering external shock, we might need to implant an internal pacemaker to their body. That whenever it detects that this happens, we then deliver a shock. Where is heartbeat regulated? It's regulated from the brainstem by the medulla oblongata. Let's have a look at the cardiac muscle cells, the action potential in there, since we kind of started talking about that a little earlier on. So we see the shape of the action potential resembling that we saw in the nerve cells earlier on when we talked about neurons. The big difference here is that we do have the same depolarization getting towards the zero. We actually overshoot a little bit. Plateau is not quite as flat as we saw in the nerve cells. It is slightly sloped, but it is still flatter or flattish. And then the return back to the normal at the repolarization. You won't notice similar undershoot refractory period as you saw in the nerve cells. The big difference here to notice is the time difference. So the action potential in a cardiac muscle cell progresses much slower. We're talking about hundreds of milliseconds in comparison to 3 to 5 milliseconds in a nerve cell. So that's the big difference that we want to notice. It takes a little long. We have done a lab where we looked at the ECG, EKG, the electrocardiogram, and that lab allowed us to become familiar that this is simply a summation of all electrical currents that we just had a look of, how they add up when you measure them externally. And there were a few elements of a trace that I wanted you to be familiar to notice: the P wave, the QRS complex, and the T wave. And all of these told about a particular event that's taking place within the heart that I want you to be familiar with, what's happening. So they tell us we would be looking from the trace, is it present, is it normal shape, and then the distances between these events from each other, so intervals of few terms that are useful for us to know when we talk about the heart. So the period of contraction, when the atria or ventricle is contracted, when the heart is contracted, is our systole. Period of relaxation is our diastole. Cardiac cycle instead refers to one complete cycle of events of the heart, so full heartbeat, complete heart. When you listen to heart with a stethoscope, you will hear two sounds, lub and dub. So it sounds a little bit like lup, lup, lup. And these sounds tell us when the valves are closing. To the first heart sound when the AV valves are closing, and second heart sound when the semilunar valves, so our pulmonary and aortic valves are closing. When we calculate the cardiac output, we do take into account the cardiac output is when you multiply the heart rate by stroke volume. Heart rate is typically around 75 beats per minute, stroke volume typically about 70 milliliters per minute. So that puts us into numbers at rest of approximately 5¼ liters per minute. But notice, if we need blood to move faster through the cardiovascular system, we can go up to 20 to 25 liters per minute. And of course, athletes can even overcome these numbers. So your heart is extremely capable of adapting to changing needs. Typically in the past, I talked with my students a little bit about congestive heart failure. We haven't really ventured there in great amounts, so I'm not gonna review it here, but you can look a little bit from the lecture video. A few words about the embryology of the heart. What's interesting, heart really early on the development, it doesn't look much more else than just a tube. And slowly this tube starts to thicken at some parts, and it starts to fold down. And eventually, very early on, we get this kind of a primitive early heart shape that's not too different from the final product. We talked about the concept that we need to bypass the lungs with the blood, so we don't direct the blood to the lungs in an embryonic heart, because there's really no need. The lungs are not functional. At this point, we have completed everything that I wanted to say about the heart, and we should review the blood vessels. And we're going to review them really kind of trying to keep up the pace. When you're reviewing the blood vessels, one thing that's useful to do is to follow the flow of the blood from the heart back to the heart. So first we have our arteries. They were carrying the oxygenated blood in the systemic circulation away from the heart. A smaller version of them, arterioles. Then we get to our capillaries. And remember, capillaries are the site where materials get exchanged. So this is the main business end. Venules, smaller versions of veins, and veins eventually carry the deoxygenated blood back to the heart. Let's have a look of what else we remember from the blood vessels. Well, blood vessels all had, in general, same 3 layers, if we look at. And we're going to move through these layers by starting from the inside of the blood vessel, and then moving layer by layer outwards from the center of the blood vessel. So let's start with tunica intima. So the lumen, just as a term, it refers to the center of any vessel. So tunica intima is in direct contact, intimate contact with the blood. It's made of simple squamous endothelial cells. So these flattened scale like looking cells if you look the cross section. So the point of these cells is that first of all, they're protecting the other layers that are going to be deeper to the blood vessel wall. So they make sure that the blood can flow quite nicely past. But also this simple single layer allows materials to be exchanged easily through this tunica intima layer. The next layer that we're going to look at is going to be our tunica media. Tunica media is the thickest layer of the blood vessels, made of mainly of smooth muscle. There is some elastin there as well. And thanks to this great amount of the smooth muscle, tunica media is able to contract or dilate, so alter the diameter of the blood vessel. So we can control the blood vessel diameter with the smooth muscle layer of the tunica media. The last layer that I want to talk about, tunica externa, sometimes called also tunica adventitia, more of in British speaking world, but you can see either one used interchangeably. So I would like you to be familiar with those. Tunica externa, really the main function of it is to support the walls, reinforce the walls, anchor the vessel to its side. So there's a lot of connective tissue there. There's also nerves and some lymphatic vessels. We did talk very briefly about the fact that just like any other tissue, the blood vessel wall will need blood supply so that it doesn't starve. So the blood supply is really easy to get for the tunica intima and tunica media. They're close to the blood flowing through. But once you get to tunica externa, that's a little bit further away. So if you have a very thick blood vessel, it might have its own blood vessel supply to the blood vessel wall. And this is known as vasa vasorum. So just something to be aware of. Let's go through arteries really quickly. So arteries, we remember they were exposed to high blood pressure and very pulsatile flow of the blood. So they're immediately after heart. So these need to be very thick. They need to be elastic vessels. We can divide arteries into a few different subcategories. We're going to start with elastic arteries. So these have thick wall, a lot of elastin in there. They're not really able to change the diameter through the smooth muscle not being that much present. They do expand and recoil a lot in response to the pulsatile blood flow. Because these don't really adjust the diameter of the vessel to control where the blood goes, they simply conduct the blood where the blood needs to go. We talk about conducting artery when we describe them. In comparison, now we're moving further away from the heart. So elastic arteries were the first arteries right after heart, with, for example, our aorta being a good example of that. Let's have a look of the muscular arteries. The majority of the arteries are muscular arteries. These have a very thick tunica media, lots of smooth muscle in the wall, less so elastic tissue. These vessels are very good at doing the vasoconstriction, vasodilation. So they're able to control where the blood goes, direct the blood where it's needed. So they are described also as distributing arteries. Of course, the smallest ones that we can have are going to be our arterioles. These are just a single layer of cells. What's important with arterioles is that they are right before the capillaries. And you remember there's much more capillaries than there is actually blood available in the body. So we can't have blood going to all of the capillaries all of the time. For that reason, arterioles have this clever adaptation of precapillary sphincters. But precapillary sphincters are muscle bands at the capillaries, either opening or closing, choosing where the blood gets directed, which capillaries are needed, which capillaries are not needed. So we open and close capillary networks as needed. Let's have a look at the capillaries a little bit. This is the business end in terms of the exchanging materials within the vasculature. So this is where materials pass across the walls. The blood pressure is very low at the capillaries. We need the blood pressure to be low because the vessel walls are so thin. This way materials can easily cross through this wall. Typically, like I mentioned, they are just a thin wall of tunica intima, so one layer only present. There are different types of capillaries, so let's have a look of those, and this could be something that I could ask in an exam. So we're going to start with continuous capillaries. These are found in skin, muscles, lungs, central nervous system, also forming the blood-brain barrier. If you look at the wall of a continuous capillary, it's really uncompromised or constantly packed, continuous layer of cells that really allows a solid, firm wall to be present. The next type, fenestrated capillaries. The term fenestration refers to a window in French, I believe. So it looks a little bit like Swiss cheese. There's little gaps missing here and there. And this allows for the materials to be exchanged back and forth. You find these, for example, in kidneys, intestines, sites where hormones are being secreted, and so on. The final example, sinusoidal capillaries. You remember sinus was something missing, a missing piece, a gap opening somewhere in the body. So these are much larger pieces missing on the capillary wall than what we saw earlier on. So we find these at the sites where a lot of exchange of materials needs to take place, such as liver, bone marrow, spleen, hormone glands, so on. The flow through these, since the wall is not as constant, the flow is much slower, which also again facilitates this exchange of materials easily. So very porous wall. We have already talked about the precapillary sphincters. The term that I don't recall if I mentioned, rather than talking about individual capillary vessels, we can just use a common term of capillary bed, capillary networks when we describe these. So hopefully that helps in terms of the language. Veins, when we talk about the veins, the blood pressure is lower within the veins. It's not quite as low as it is in capillaries, so lowish, if you wish. Majority of our blood vessels are veins. The big thing here that we talked about was how do you get blood back to the heart, for example, against the gravity. And this was all thanks to a phenomenon known as venous return. So veins have a thin wall, so they easily get squeezed by skeletal muscles all the time as we move, but also there are some squeezing, for example, when you breathe, those respiratory movements squeeze a lot of the veins within your thoracic cavity. So as the vein gets squeezed, the walls of it get squeezed, and the blood gets pushed upwards towards the heart within the veins. Well, when the squeezing stops, we don't want the blood to return back to against the gravity, so we have valves present in veins. So same adaptation that we saw in the heart, facilitating unidirectional flow of blood. So this stops the blood from returning backwards and always keeps pushing the blood towards the heart. The time when this would not work is, for example, if you have a soldier who's standing on a parade with knees locked, not moving. What we do find sometimes they're not getting enough blood then returning to the heart, and from the heart, of course, to the brain, and then as a result, they might pass out. So that would be a good example where the venous return has not been fully facilitated. The term anastomosis referred to these interlinked interconnections of the blood vessels, collateral circulation, if we have alternative routes for the blood to pass onwards. Let's look at a little bit of the physiological aspects of the blood vessels. And we're going to start by talking a little bit about the blood pressure. So blood pressure is simply defined as the force that the blood exerts against the wall of the blood vessel. And what you'll notice that typical values are usually 120 over 80, 120 over 90. There is an upper and there is a lower value. This is because we will measure the blood pressure during systolic and diastolic times, so during the contraction and during the relaxation, measured in millimeters of mercury. There's many factors that play a role when we look at the blood pressure, how much blood you have, of course, how hard your heart is putting that blood out, the resistance for the blood flow, vessels, whether they're very elastic or not, or so. I mentioned that my blood pressure tends to get a little higher when I go to see a doctor and a nurse. That is known as white coat syndrome. You get a little nervous, so that's a normal reaction. So blood pressure is typically a little lower when you measure it at home at your own pace. We also did a complete lab looking at the pulse. So I won't go into too much of a detail of that, but it is an important method for us to get information about the function of your heart, including, for example, the heart rate. So whenever the ventricles push the blood out of the aorta, the blood pressure goes up. And whenever the ventricles relax, the blood pressure goes down. And it is this wave-like pulse that we can feel in various spots where we might measure the pulse from. And there's many locations. We had a look of these also at our lab. So carotid pulse from the neck, where the common carotid divides into internal and external. Ulnar pulse from the ulnar side of the wrist. Radial pulse on the radial side of the wrist. Brachial plexus pulse from the cubital fossa. Our popliteal pulse at the legs, posterior tibial and dorsal pedis pulse from the feet. Femoral pulse, really good, strong to measure from the groin, but obviously we don't tend to do that unless we have no other option. That's a kind of a little private area. From the temple of your head, superior temporal pulse and external maxillary pulse, you sometimes can feel that really blood vessel. You might even see it popping when someone has exercised hard. We talked about the regulation of the blood pressure, and in short-term regulation, we used our good old diagram of basics of understanding the homeostatic mechanism loop. So we went into much more detail at that time, so this is going to be just a quick run-through. So if the blood pressure changes and it's off from the set point, which would be 120 over 80, I think I haven't updated this graph, we compare it within the medulla of the brain. So we want to see if the blood pressure where it should be. Well, how this was detected, it's through sensors at the baroreceptor or chemoreceptors. These are located, for example, at the carotid sinus, so where the common carotid divided into external and internal carotid, there's an expansion there. That's where these reflexes are, these sensors are. Also leaving from the heart at the aortic arch, we had receptors there detecting if the blood pressure is what it should be. And same goes for most of the large vessels. So we're constantly comparing the current blood pressure against what it should be, what the set point is. And this happened at the medulla. If the blood pressure isn't where it should be, we can do many things. Of course, medication is what we doctors tend to prescribe. Treating the cause is really important. And we do have an option as immediate reaction to also do vasodilation, vasoconstriction as needed. Long-term regulation of the blood pressure is through the kidneys. We can change the blood volume, which then would change the blood pressure. We have a direct mechanism, which is through the kidneys, and indirect mechanism, which is more of a complex, known as a renin-angiotensin-aldosterone mechanism. And you can find as much as you need of that from the lecture video. So really, to sum up, we want to have high enough blood pressure so that the tissues get enough blood, so that there's adequate tissue perfusion, but we don't want the blood pressure to be so high that the blood vessels get burst or damaged. So it is a delicate balance of between the two that we want to have. Some clinical considerations, we should look at high blood pressure, so hypertension. This has many possible consequences that are negative: heart failure, vascular disease, kidney issues, stroke, and so on. And many factors contribute to this. Rarely we can point out just one factor. It is typically multifactorial. There are certain lifestyle changes that you can do to improve. And of course, we do have medications, blood pressure medications, that quite a few people end up taking, especially as they age. Let's have a look at hypotension, low blood pressure. In low blood pressure, we do not know whether it really is as big of an issue as the high blood pressure. Some research recently has suggested that might be beneficial. There's many causes that it can be just a temporary, for example, when you stand up, that can cause your blood pressure to be low temporarily. It can be chronic. Usually chronic low blood pressure is due to poor nutrition, or there can be an acute reason, such as you going into shock. And let's have a look of the shock a little bit more. So I wanted you to know the signs and symptoms of the shock. And the mnemonic that I gave you for doing so was PC FATS. So individual who's about to go to this shock, the skin often looks pale. There's not much blood flowing there. And for that same reason, it feels calm, kind of clammy, cold. The breathing is typically fast and shallow, so very much like same for the pulse, that is fast, weak. These people are, it's not comfortable. They are getting quite agitated, so you might want to keep that in mind if you do first aid. Don't get on the line of getting punched. They usually are very thirsty as well. These are all signs and symptoms that the individual might be going into shock. Important in terms of immediate treatment, we do not want these people to collapse and hit their head, so we tend to sit them down or lay them down. We really want to treat the cause, but in the meantime, we want to make sure that there's an adequate blood supply to the brain. So really raising the feet, laying them flat is ensuring that enough blood is flowing to the head part. Blood flow regulation, extrinsic, intrinsic. And then we talked about many different special circulations that we had. So we had local factors, local reasons for having the blood flow control to those sites. So we discussed that more in detail at the lecture videos. Then I have provided you a bunch of diagrams showing the various circulations, various important blood vessels, arteries, and also veins. What I do want to talk before we wrap up the portion on the blood vessels, I want to talk about the speed of flow of the blood. So let's have a look of that a little bit. What we'll notice that the speed of flow of the blood is lowest at the capillaries. So that makes sense because it was within the capillaries where we wanted to exchange the materials. So if the blood would move too fast, we wouldn't have time for this exchange to happen. Well, one good question in an exam would be that, okay, well, since the speed of blood is lowest at the capillaries, why is it? What's the factor that contributes to the speed of blood in these different vessels? Well, that really is the total area of the vascular bed. And you'll notice that the total area of the vascular bed of capillaries is the largest. So relatively, as the area grows, the speed of flow decreases. You'll also notice that the speed of blood, even though it drops when we get to the capillaries, it picks up a little bit when we return away from the capillaries or continue our way away from the capillaries. Could make a good exam question, just putting it out there in the universe. We didn't talk at this point too much about hydrostatic and osmotic pressures. I saved it for when we talked about the lymphatic system. But the key thing was that there's forces pushing blood out of the blood vessels and forces pushing blood back into the blood pressure. If these two were in a complete balance, we would never lose any plasma from the blood vessels. We knew that that was not the case. We are actually left with a little bit of the plasma leaking out in a day, so that needs to be picked up some way, and that was the lymphatic system that stepped in to picking that up. If we were not going to pick that up, we would end up buildup of fluid outside the blood vessels at the interstitial space, and that would of course cause an edema, so that would be a problem. A couple of key points just to summarize what we should know about the blood vessels. We do typically have arteries run deeper than the veins, so they are a little bit more protected in that sense as well. Cutting a major artery could cause a death rather fast when we lose a lot of blood. Veins are thinner walled, so they're usually a little more superficial. We had many alternative roads, so that those were the anastomosis. And that brings us to the very last section of our review. So having a look of the lymphatic system, and in the interest of keeping us going and being mindful of each of our times, I'm just going to jump through that, go through it probably a little faster. You have the full lecture videos, and we just covered this last week to refer back to. So we remember that all of the circulatory, lymphatic, and immune system, they were all linked together. And really this chapter, I think, brings home that concept rather well. So we remember the balance between hydrostatic and osmotic pressure left at little plasma leaked out of the blood vessels every day. That plasma is exactly same as the interstitial fluid in its composition. So we're talking of the same stuff, but since the interstitial fluid is no longer within the blood, it's now in this interstitial space, we call it with just a different name, interstitial fluid. When this interstitial fluid gets picked up by the lymphatic vessels to be taken back to the circulation, now we talk about lymph. It is still this very same stuff, but when it gets picked up by the lymphatic vessels, when it enters the lymphatic system, we start talking about lymph. Another excellent exam question to be aware of. Where does the lymph interstitial fluid become a lymph? Well, when it enters the lymphatic system. If we compare lymphatic system and cardiovascular system, both were one-way systems, so the flow of material was to one direction and one direction only. The difference is that unlike cardiovascular system, which is a closed loop, lymphatic system is open from one end, so it picks up the fluid from one end and carries it and then dumps it close to the heart. The functions of the lymphatic system, there were three that I wanted us to know. I think the two first ones are more obvious ones. So of course we're fighting against pathogens. That's where the white blood cells became very important. So pathogens were any external invaders to our body that shouldn't be there, can cause trouble, can cause disease. So our lymphatic system, in collaboration with the immune system, play an important role in that. You can think of lymphatic system almost like the housing of the immune system. The other role is, of course, this fluid transport. We're transporting this extra interstitial fluid back to the bloodstream. Otherwise, if we didn't transport it, we ended up with lymphedema. The last function that a lot of time we don't get to think about, we'll see it when we talk about a digestive system a little bit more, is that sometimes we have molecules such as large fat molecules, but it could be other things as well that are too large to be picked up by our capillaries of the cardiovascular system. Well, lymphatic system capillaries are able to pick larger molecules, so we can recruit that to pick those up and transport them to the cardiovascular system when the lymph gets dumped into there. In terms of the lymphatic vessels, we have the same capillaries and then growing and growing in size as we move along the lymphatic vasculature. So we're going to go through those quickly, looking at them in an order. So everything started at the capillaries where it picked up the lymph from the interstitial fluid. So let's have a look of the lymphatic capillaries. They are open-ended, so they are not a closed loop like cardiovascular system was. So they're picking up this interstitial fluid around the cardiovascular system capillaries where it leaks out. They are more permeable, even more permeable than the blood vessel capillaries. Made of endothelial cells, simple squamous endothelia, so it is this kind of flattened like disc, so it again facilitates easy passage of materials across there. The next biggest vessels that we end up seeing are our lymphatic vessels, so they are collecting vessels. They are thin. The wall of these vessels is thin at the lymphatic system. They do have the valves similar to the veins that we saw in the cardiovascular system, facilitating this one-way flow of blood. And we do have a lot of anastomosis, these interconnections between lymph vessels. Two that I mentioned were in the thoracic cavity, running at all the way at the back, the thoracic duct, and at the inferior portion of that dilation sternoculi. So that was in our thoracic cavity, running back there. Lymphatic trunks, a little bigger vessels again, so located, named based on their location. Lumbar trunk at the lower back, at the lumbar region of the vertebral column, intestinal, covering the intestines, intercostal, of course, between the ribs. Bronchomediastinal is a good term, tells us that it drains the bronchia mediastinum, subclavian, of course, below the clavicle, and jugular, the neck and throat region. In terms of the ducts, so a little bigger as well, too, that I wanted to share, a right lymphatic duct draining the right arm, right head, and right neck, emptying into the right subclavian, and then the thoracic duct draining the remaining parts and into subclavian as well. Let's have a look of the lymph itself. What is lymph made of? So we find the same things that we found in the plasma. So water and proteins, also salts, glucose, and fats. The difference to the plasma is that we now have, of course, white blood cells there. So these cells of the immune system. One thing that you should never see in the lymph is going to be our red blood cells. If we see red blood cells there is the problem, and we need to be a little concerned about that. Where is lymph produced? At the bone marrow and lymph nodes. The clinical relevance of that is that when the lymph builds up and doesn't get picked up, we are left with swelling, so edema, lymphedema in this case. This can be caused by tumors, the removal of lymphatic vessels, and so on. So typically, spontaneously, some amount of a recovery from that can take place. Let's have a look at the lymphatic cells. So we have the immune system cells, so B and T cells that are important. T cells named after maturation site of thymus, B cells named after maturation at the bone marrow. Then we have supporting cells. We didn't really get to have a look of those too much. Let's talk about lymph nodes. These are these small, about 1 inch sized, kind of bean-shaped glands, and they are typically located as clusters, so around large lymphatic vessels. So one way to describe them has been that I've heard is that they're almost like a pearl necklace. I think you need to have a little imagination to see that, but let it be. There's hundreds of them in the body. Their function is really twofold: to filter the lymph that goes through, and that's where the immune system cells also are housed, so when they are needed, so we can fight pathogens. You remember when there was an inflammation, the lymph nodes do tend to swell. So the white blood cell numbers increase within the lymph nodes, and physically the lymph nodes swell, they get a little tender. The locations where we can find lymph nodes, again named based on the body location: cervical lymph nodes at the neck, axillary lymph nodes at the armpits, supratrochlear above the trochlear notch, inguinal, the groin region, abdominal and thoracic, well, abdomen and thorax respectively. If we have a look of the parts of the lymph nodes, they do have a cortex and they do have a medulla, as we've discussed before, within the medulla sinuses, so openings within there where the macrophages live and they screen the lymph. Lymphatic vessels, we have vessels coming into the lymph node. These vessels are known as afferent, and we have lymphatic vessels leaving the lymph node. These are known as efferent. The swollen lymph nodes, we talked about that, how doctor will really look at that as a sign. If the lymph nodes are swollen, they're tender, it is likely that there's a lot of white blood cells there. This is in response to an infection having taken place of somewhere in the body. We might not know where it is, but we already have a clue that infection is taking place. Let's have a look of the couple of important lymphatic organs that we want to know, we want to discuss about. And we're going to start with the spleen, which is about fist-sized, really the largest lymphatic organ, located at the left side of the abdominal cavity, just below the stomach. This receives a rich blood supply. So the group that did the diagram of the abdominal region of the vasculature, they hopefully added a lot of blood supply to the spleen, splenic arteries, splenic veins, so there's a rich blood supply. The function of the spleen, there's many. It's where the immune surveillance, immune responses take place. They do clean, the spleen does clean the old dead blood cells, platelets, and so on away. It also stores iron. Platelets can be reused. In embryo, early life that you have, it is the major site of the red blood cell production as well, the spleen. So something to keep on. We have talked a little bit about tonsils. So there's actually a bunch of tonsils, not just one set of tonsils that we often think of, named based on their location. So palatine tonsils are the posterior oral cavity tonsils, the ones that we typically think of. But there are tonsils also at the base of tongue. These are lingual tonsils, tonsils at the posterior nasopharynx, so pharyngeal tonsils, and at the opening of the auditory tube to your nasal cavity. So these are our tubal tonsils. If you have an inflammation of tonsils, it's going to be tonsillitis. We can remove them surgically in a process known as tonsillectomy. We used to do this almost as a kind of standard response. These days, we're a little more careful because we are realizing that tonsils might play a bit more of an important role in the immune system. Appendix is the first part of the large intestines, kind of hanging out from the cecum before we get to the colon. So this little hanging bit after small intestines. Appendix can get inflamed, so it can get removed if it's inflamed because we don't want it to burst. So the question would be, why do we have an appendix then? Well, our best theories at the moment suggest that that's an evolutionary remain from when we ate a much more plant-rich diet. So that might be the explanation there. That's where the bacteria to digest that were housed. I do want to talk a little bit about thymus as well. Thymus is the structure that we've seen before, large in your infancy and in your puberty. I think it's replaced by connective tissue and fat tissue, site where the T cells mature. So that really kind of wraps up everything I wanted to say about the lymphatic system, and we have left to look at the immune system. So let's talk about this functional system. It is not really so much of a structural system. So it really kind of free rides, is located within the lymphatic system in terms of the structures, but much more functional in nature. So it's the white blood cells that we're interested here, how they work and what they do. Their main function is to defend us against pathogens, so any disease-causing virus, bacteria, parasite, fungus, there's many things that can cause us to get sick. They fight against those. What pathogens typically do is that pathogens tend to invade your body and to multiply in numbers to cause issues. We had few branches for the immune system: innate immunity, which was much more about general responses. They protect against many different kinds of pathogens. And innate immunity worked through few different lines of defense. The first line of defense is to form physical barriers, such as having the skin blocking the pathogen from getting in. So skin cuts are that bad for that reason, that pathogens can get in. Any puncture in the skin is a route for potential infection to get in. We don't only have skins, of course, we have mucous membranes, the more internal barriers that we've talked about. Second line of defense, various chemical responses such as sweat, stomach acids, tears, rinsing the urinary tract. As we urinate, urine was sterile, so that helps as well. So kind of if we cannot keep the bacteria, can keep the other pathogens away by forming a barrier, then let's at least kill it chemically. If that fails, we always have an option of recruiting our cell responses. And you can look more of those into lecture video. We did talk a little bit about inflammation, signs and symptoms of inflammation, and I think that that would be handy for you to know. So let's look at that a little bit. If a site is inflamed, you typically notice it is kind of a reddish in color. It is kind of warm because there's a lot of blood flowing there. So we're really bringing those white blood cells there to deal with that inflammation. This causes also, because there's so much stuff there now, swelling as well. These sites are painful. This helps us not to use it, and also be careful with these structures. The fifth characteristic that I want to add is loss of function. In the inflamed site. So all of these 5 are really good signs and symptoms of an inflammation. I think I do ask that in the exam as well. Fever refers to abnormally high body temperature, and we discussed that it might be beneficial to have a fever for recovering, getting healthy. So maybe we're over-medicating a little bit on that, of not letting people to have a fever that might be beneficial for their recovery. The second part of the immune system that we had a look of was our adaptive immunity. We call it acquired in UK. I think that that's a little bit better. It explains how you meet the pathogen first, and then you develop a response to that memory against it. So if you meet it on the second exposure, you have a very targeted, very specific response against a specific type of antigen. And one big thing there to remember, where we talked about immunoglobulins. These are really the workhorses. I did list some examples when immune system goes wrong, when immune system can be compromised, when your immune system is overly active. All of those you can refer back to in our actual lecture videos. And I think that that really completes our review of the study guide for the exam 2, where we had a look of the chapters on the heart, on the blood vessels, and on the lymphatic and immune system. I know that it's a lot of information, but remember, I do facilitate open book tests. It is time though, so you do need to know the material. You can't just be looking it up. And we are not going to do them in a proctored format, so it's just you can do it wherever you wish. Log in. It is a single sitting, so you do it on one go. That about completes everything that I had planned to say. Let me just jump back into another view, and I'm gonna share with you the PowerPoint just as a reminder that I do happily answer if you have any questions. All you have to do is reach out and I'll make time to make the time that works for everyone. If you have anything about the chapter contents or anything about practical issues. Quick shout out, do remember to get on top of those homeworks as I will be closing them at the end of this week. If you run into any technical issues while taking an exam, let me know and I can definitely reset it for you. That completes everything I had planned for today. I appreciate you sticking around and I will either see you later tonight or then tomorrow, or if not either one of those, then the next week when we move to new lecture materials. Thank you and have a very good rest of the day.